Welcome to this explainer. Today, we're focusing our attention on one of the most highly automated, strictly regulated, and honestly, utterly fascinating microscopic operations in existence, the cell cycle. I want you to imagine, if you will, a microscopic factory that literally never sleeps, constantly working to perfectly replicate itself. Well, that's the incredible reality happening inside you right now.
So, when you really think about it, it all boils down to this fundamental biological question. How does one single cell safely and accurately become two? How does it know when to grow, when to copy its vital instructions, and exactly when to split apart without making a catastrophic mistake? The answers actually lie in an incredibly precise sequence of events.
Okay, let's dive into this overview map. As you'll see, the eukaryotic cell cycle is broadly divided into two main chunks. First, you've got that large blue and green area taking up most of the cycle. That's interphase. That's where the cell spends the vast majority of its time growing and preparing. Then there's that much shorter but absolutely crucial yellow section, the mitotic or M phase, and that is where the actual physical division takes place.
Here's our roadmap for this explainer. 1. The cell's daily life. 2. The big split. 3. Quality control checkpoints. 4. The molecular supervisors. And 5. When control fails.
All right, let's kick things off with Section 1, the cell's daily life. Let's step right onto the factory floor into the main operational state, interphase. Now don't let the name fool you. This isn't a resting phase at all. It's a period of intense, methodical preparation. It starts with the G1 phase, or the first gap. Here, the cell is physically growing in volume, synthesizing the lipids, carbohydrates, and proteins it needs. It's basically gathering all its raw materials. Next up is the S phase, which stands for synthesis. This is arguably the most critical step, because the cell is carefully replicating its entire DNA blueprint, forming two identical sister chromatids attached at a centromere. Finally, it enters the G2 phase, or second gap. Here, it's making final preps. Mitochondria replicate to boost energy, the centrosome duplicates, and microtubules form so they can develop into the spindle fibers that are going to be absolutely necessary to pull everything apart later.
And this brilliantly illustrates something super important. The cycle isn't just an endless loop for every single cell. There's actually an off-ramp built into the system. Not every cell just automatically continues dividing.
We call this off-ramp the G0 phase. You can think of it kind of like the factory's break room. If a cell during the G1 phase realizes, hey, I have insufficient energy or nutrients to continue this demanding cycle, it just exits into this non-dividing state. And the really cool thing is, while some cells stay here permanently, for others, this exit is completely reversible if conditions happen to improve.
Moving right along to Section 2, the big split. Assuming our cell has passed through interphase perfectly, the factory shifts gears into active, physical division. This starts with mitosis, which is specifically the division of the cell's nucleus, sometimes called karyokinesis. Mitosis is a highly choreographed performance broken down into 4 distinct stages: prophase, metaphase, anaphase, and telophase. Across these 4 stages, the duplicated chromosomes get organized, aligned perfectly in the middle of the cell, pulled apart to opposite poles, and finally enveloped into 2 brand new nuclei.
But hey, dividing the nucleus isn't the whole shebang. We still have to divide the rest of the factory, the cytoplasm itself. We call this cytokinesis, and it's fascinating how different biological factories handle this mechanical finish. In an animal cell, a cleavage furrow forms. Basically, a contractile ring made of microfilaments literally pinches the cell membrane from the inside inward, kind of like pulling a drawstring tight until it snaps into 2 separate cells. Plant cells, on the other hand, have rigid cell walls. They can't pinch, so instead, vesicles containing cell wall materials from the Golgi apparatus gather in the middle and build a literal dividing wall called a cell plate. It just builds from the center outwards until the 2 new cells are totally walled off.
Which brings us to Section 3, quality control checkpoints.
Now, what's really interesting about this slide is seeing exactly where the security cameras, so to speak, are placed on our map. With a process this complex, a factory needs a strict security system to avoid churning out damaged goods. So the cell features 3 major checkpoints: G1, G2, and M. These are literal stop signs built into the cycle. The cell completely halts at these specific points and absolutely will not proceed until it gets a definitive go-ahead signal.
So let's look at exactly what these security checks actually entail. At the G1 checkpoint, the cell inspects itself to ensure adequate size, sufficient nutrients, and confirms there is absolutely no DNA damage before it commits to copying its blueprint. Later on, at the G2 checkpoint, it verifies that DNA replication completed successfully and double-checks again that the newly replicated DNA isn't damaged. Finally, right in the middle of mitosis, the M checkpoint ensures that every single chromosome is securely attached to the spindle microtubules from both poles. Only a completely perfect inspection allows the factory line to keep moving forward.
Next up, Section 4, the molecular supervisors.
Okay, so we know where the checkpoints are, but who's actually conducting the inspections? Well, your factory supervisors are two specific types of regulatory molecules. The first are cyclins, which are proteins whose concentration cyclically fluctuates inside the cell. The second are CDKs, or cyclin-dependent kinases. These are enzymes that actually remain inactive until a cyclin comes along and binds to them. When they finally bond together, they form a cyclin-CDK complex. These complexes are the literal molecular keys that unlock the checkpoints. They act as the vital go-ahead signals.
Take the G1 checkpoint here as a prime example. If the cell's internal supervisors determine everything is correct, the cyclin-CDK complex provides a green light. Getting this go-ahead signal here essentially commits the cell to division. It's going to go on to complete the S, G2, and M phases. But if the inspection fails, it gets a red light and is diverted right out of the cycle into that non-dividing G0 resting state we talked about earlier.
And finally, that brings us to Section 5, when control fails.
So, what happens if this beautiful, intricate system completely breaks down? Well, the crucial point is this. Cancer is fundamentally a disease of the cell cycle. It happens when a normal cell becomes abnormal and somehow manages to bypass these crucial inspections. Normally, your immune system recognizes a damaged cell and destroys it. But if it manages to evade destruction, it continues to divide in a completely unregulated manner. These cells don't care if they are densely packed together, and they don't even stop dividing when growth factors are completely depleted. The factory safety brakes have simply vanished.
Now, this unregulated division leads to a mass of abnormal cells called a tumor. But we're looking at two very different outcomes here. A benign tumor means those abnormal cells are remaining trapped right at their original site. However, if those abnormal cells gain the ability to spread to new tissues and start impairing the functions of one or more organs, well then, it becomes a malignant tumor. And an individual with a malignant tumor is said to have cancer.
As you can see in this stark visual, in a benign tumor, the mass is totally contained, just wrapped neatly in its original location. But a malignant tumor is aggressive. The abnormal cells literally break out, invading the surrounding healthy tissue, totally ready to spread their unregulated division elsewhere in the body.
Which really leaves us with a pretty profound realization. By studying something as incredibly tiny as the growth phases, the spindle fibers, and the cyclin proteins of a single microscopic factory, we are actually studying the very foundation of major disease. Could mastering our understanding and control of these tiny molecular checkpoints eventually be the ultimate key to defeating cancer once and for all? It is an absolutely fascinating thought to leave you with today. Thank you so much for joining me for this explainer.